Hello, and welcome to my talk. My name is Nicole Shu, and I'm a postdoctoral scholar at Caltech with John DeBerry. Today, I'll be talking about field testing of biohybrid robotic jellyfish to demonstrate enhanced swimming speeds. This is work that my colleagues and I conducted at the Marine Biological Lab in Woods Hole, Massachusetts last year. To motivate this work, the ocean is an important resource. We know that the ocean helps regulate the climate, modulate oxygen and carbon dioxide levels, and provide goods like food, medicine, and transportation. But even though the ocean makes up over 70% of the world's surface, over 80% of it is still unmapped, unobserved, and unexplored. The current tools we have for monitoring the ocean include remotely operated vehicles, or ROVs, and autonomous underwater vehicles, or AUVs, like the glider shown on the right. These have great advantages and can travel fast speeds over long distances for data collection, such as ocean mapping or deep sea exploration to uncover new animal species and behaviors. However, many of these technologies are still limited. The disadvantages include being costly, bulky, consuming high power, and leaving larger vehicle wakes that might disturb more sensitive areas of the ocean among more delicate animals. As an additional tool, we can look to nature for inspiration to build bio-inspired aquatic vehicles, which can leave more natural wakes to reduce these environmental disturbances. These vehicles can approach more delicate areas of the ocean, like among the coral reefs. Two examples shown here are the aqua AUV and soft robotic fish. Now, each particular model organism offers its own advantages and disadvantages, but today I'll discuss why jellyfish are an ideal model organism. Despite over 500 million years of evolutionary pressure, the body structure of the moon jellyfish has remained largely unchanged. It's been speculated that this is because of their high energy efficiency, so they're useful to study for underwater vehicles because they have a low cost of transport, which is one metric of energy efficiency. The cost of transport is defined as the energy per mass per distance traveled, so lower values equate to higher energy efficiencies. This plot shows the cost of transport for a variety of animals, as well as AUVs, with log-scale axes. As shown, jellyfish have the lowest cost of transport compared to other animals and AUVs, so we might learn how to make more energy-efficient robots by studying how jellyfish swim. But there are various approaches to bio-inspired designs. On one end of the spectrum are purely mechanical robots, which have well-controlled inputs and outputs, but they often require a lot of power and are still connected to external batteries. On the other end are biological constructs, like this medusoid made from seeding rat heart cells onto a silicon base. This is proof that we can use muscles as actuators within robots, but the cells are limited to specific media for survival. The approach I'll discuss today is to build a biohybrid robot by integrating microelectronics onto live animals. This is a low-power and low-cost strategy and is adaptable to many environments because of the ubiquity of natural jellyfish in the ocean. To start, let's dive into the swim anatomy. On the left is a bottom view of a jellyfish, and in each of these 8 circles is a pacemaker, or a sensory organ that innervates the swim muscle. The swim muscle is a ring oriented on the underside of the jelly, shown here in red, and even if only one of the pacemakers fires, the entire muscle ring is eventually activated. To swim, the jellyfish activates any or all of its pacemakers, which then causes the muscle to contract during the power stroke and relax during the recovery stroke. Instead of relying on the animal itself, we can use electrodes to act as artificial pacemakers to activate the muscles directly using an electrical signal, as shown here, with the voltage plotted over time. These are off-the-shelf components for a self-contained swim controller, including the housing and cap for waterproofing, the microcontroller, battery, and 2 electrodes for straight swimming. This is what the system looks like once it's put together, and you can see that the electrodes have LEDs for a visual confirmation of the electrical signal. The swim controller embeds into the jellyfish as shown here. The animal is in white, the swim controller is in blue, and is attached using a wooden pin shown in yellow into the animal, with the electrodes embedded into the tissue near the margin shown in red. This is an example of what the biohybrid robotic system looks like in the lab. You can see that the jellyfish with the swim controller attached is able to swim strongly against the background current, as opposed to the other jellies in the tank that are primarily being carried by that current. The swim controller also does not harm the animals, and for more information about that, I've written a paper with bioethicists on the ethical considerations of this work. You can find the preprint available at this DOI, or follow the QR code. To determine how externally driving the swimming frequency affects swimming speeds, I conducted a series of experiments in the laboratory in a 2-meter tall tank, with the jellyfish initiated from the top of the tank swimming downwards. From these experiments, we found that we can increase jellyfish swimming speeds up to 2.8 times their baseline. This plot shows the enhancement factor, which is defined as the stimulated speed over the unstimulated or baseline speed versus the swim controller frequency. We found that the enhancement depends on the body morphology or shape as defined by the fineness ratio, which is the ratio of body height to diameter. So, flatter, more oblate individuals didn't have much enhanced propulsion, but rounder and more prolate animals did, which shows that we can choose more prolate animals for faster robotic control. For more information on the laboratory experiments, you can refer to my paper published earlier this year. But these laboratory experiments were all conducted under quiescent controlled environments, and if our goal is really to use these biohybrid robots in the ocean, then we certainly won't have quiescent or controlled environments there. So the question is, how do these results compare when biohybrid robotic jellyfish are tested in the real world? To answer this, my collaborators and I traveled to the Marine Biological Laboratory in Woods Hole, Massachusetts. Back when we could travel and do things. This is a map of Massachusetts, and if we zoom in on this red star, we can see Woods Hole, as well as us in happier times when we were able to interact with each other in public. So this is where Jack, Sean, and James went scuba diving for these experiments, and I was on shore with the jellyfish and extra swim controllers. Here's a schematic of the coastal conditions. One scuba diver imaged the biohybrid robotic jellyfish as it swam upwards at various frequencies, and there was a rope with alternating yellow and red markers so that we could calculate the location of the jellyfish relative to a ground truth in the background. And this is an example of what the experiments look like. You can see the jellyfish in frame swimming upwards, with the rope, that's that white vertical line in the background, and fish and other marine life are able to swim around the biohybrid robotic jellyfish. So from videos like these, we could then plot the vertical displacements over time, and by plotting those vertical displacements over time for different frequencies, we can then compare to previous laboratory experiments by calculating the enhancement factor. So this plot shows the previous laboratory data in gray, with different markers representing the 6 different jellies in the lab. The field data using 2 animals is plotted here in red and blue, and we can see that the enhancement of swimming speeds in coastal conditions is comparable to laboratory results. The absolute swimming speeds increased from 2 centimeters per second in the control cases at 0 Hz up to 6.6 centimeters per second for jellyfish 1 when driven at 0.75 Hz. There is high variability at 0.75 Hz, and that's caused by the presence of endogenous contractions because we're not arresting the animal's natural behavior. And we can talk more about that if you're interested later. In addition to the field experiments, we wanted to see whether we could predict the jellyfish swimming speeds using a simple hydrodynamic model. So starting from Newton's second law, I modified a mechanistic model that incorporates body kinematics in thrust, drag, and acceleration reaction forces. The only inputs were the animal's geometry, such as the bell height and bell diameter, as well as time-dependent parameters like the contraction and relaxation times. Using these simple inputs, this plot shows the theoretical vertical speed over the experimental speeds tested in the field. Even though this hydrodynamic jetting model neglects jellyfish paddling behavior, it's still able to predict swimming speeds with a mean error of 0.8 centimeters per second. So for future robotic control of jellyfish, if we want to use faster animals, then we can input its morphology into the model and predict what swimming speeds we'll get a priori. So to conclude, robotically controlled jellyfish in coastal conditions can swim up to 3 times faster than natural jellyfish, from 2 to 6 centimeters per second, with comparable enhancement factors to previous laboratory results. A hydrodynamic jetting model can accurately predict jellyfish swimming speeds using morphological and time-dependent input parameters from the real jellyfish. And we've shown a proof of concept that biohybrid robotic jellyfish can work reliably well in field conditions. In the future, we can work to improve the maneuverability by incorporating turning and closed-loop feedback. We can also add sensors and improve the overall robotic design by using biodegradable electronics or more advanced batteries. This is in the hope that in the near future, we can deploy swarms of biohybrid robotic jellyfish in the ocean to take important biogeochemical measurements and track markers of climate change. With that, I'd like to thank Cabrillo for providing us with jellyfish, the MBL and my collaborators, the Dabiri Lab, especially Valerie and Jen for shipping live jellyfish across the country, and our scientific illustrators and funding sources. If you'd like more information, please see the preprints of the fieldwork and ethics papers, and you can contact me here. Thank you very much for your time and attention.